All right, so I don't have a very loud speaking voice. Can you hear me if I speak into here? Okay. And then if you can't hear me at some point during the talk, just start waving and I know what that means. Okay, so today I thought I would actually focus on a couple of my postdoc projects. So I did my postdoc in embryonic tendon development with Ronan Schweitzer at Shriners Hospital in Oregon. And so what I really want to do today is maybe not kind of throw a whole bunch of data at you, but to really present the heart of these two stories. So the first is on the unique development of the superficial flexor muscle. And the second is on a new conceptual model we've established for understanding tendon growth and elongation. And then finally, just in the last few minutes or so, I'll focus sort of on the direction I'm going to take my lab, where I take some of the strengths from my postdoc and PhD research and apply it to looking at stem cell biology and tendon healing. So the musculoskeletal system is obviously a system composed of several different tissue types: muscle, tendon, ligament, and bone. And so proper function of the entire system requires not just the proper development and differentiation of each of these tissues in isolation, but really also the proper connections that are formed between them during development. And so while the Schweitzer lab, my postdoc lab, focused on tendon development, what we found was that to understand how tendons develop and differentiate, you really couldn't separate it from its adjacent tissues. And so there's a great degree of interdependency between tendon development and that of muscle and skeletal development. And so one of the central questions was how the musculoskeletal system really comes together during development. So compared to muscle or skeleton, there's actually very little still that is known about the factors that regulate tendon cell fate. And so one of the first transcription factors that was identified for tendons is Skeraxis. And so Skeraxis labels all tendons and tendon progenitors from early stages onward. And so when you look at tendon development and Skeraxis induction in the limb, you can see that so the limb forms by these initial budding at E9.5. And then the limb is formed by progressive or proximal to distal progression of the skeletal parts. So first you have formation of the humerus, and then the forearm, and then the autopod or the hand. And you can see at these stages, Skeraxis induction also progresses in this proximal to distal fashion, where you have an early induction at E10.5 and then progressive induction of the elements as they are formed. However, it's really not until E12.5 that something like a tendon pattern or recognizable tendon pattern emerges. And so for my talk, I'm really going to focus a lot on kind of these stages between E12.5 and E14.5, because we find that these are really important central stages for how the musculoskeletal system begins to organize itself. And so because tendons are these long, thin structures that have complex trajectories and insertions, the best way we found to analyze tendons was by transverse sections taken all the way through the limb. And so this is the kind of data that we generate, which is not intuitive and makes no sense to other people but us. So for a lot of my talk, I'll be focusing mostly on these schematic representations of the data. Using a ScxGFP transgenic tendon reporter, we can very easily visualize tendons in these sections, which are shown as green. And then if we stain for muscle-specific myosin, we can see other tissues like muscle, or stain for other things. All right, so this is more for the basic researchers in the room, but just a little review of the muscle anatomy in the limb. There are generally two groups of flexor muscles. Muscles that are extrinsic to the hand, so located completely within the arm, and these muscles are attached to long tendons that insert somewhere within the hand. And then there's a second category of intrinsic flexor muscles, and these muscles are localized completely within the hand, along with their short attaching tendons. And so the superficial flexor and the profundus flexor are examples of these large extrinsic muscles, and then muscles like the lumbricals or the interosseous muscles are these intrinsic muscles. And so by embryonic day E16.5, all of the muscles and tendons in the limb are fully patterned and formed, including the superficial flexor, which is shown here in orange. What is interesting, though, is if you look 2 days before that at E14.5, all of the other tendons are formed, and the profundus is shown here in blue as an example, but if we look for the superficial flexor, we actually see that the digit parts are missing. In fact, most of the superficial flexor tendons are missing, except for this small fragment here at the metacarpal phalangeal joint. And then instead of these long proximal tendons, these little tendon fragments appear to be attached directly to 3 intrinsic muscles. So while in humans there are 4 superficial flexors, in mice there are only 3. So this was surprising, and so because these muscles were directly attached to what seemed to be superficial flexor tendon fragments, we thought, well, maybe they could be the superficial muscles. However, I just explained that the superficial muscles are these extrinsic muscles that are located in the arm, right? And so alternatively, they could be maybe transient lumbrical muscles that just appear and then disappear by the end of development. And so confronted with this, the first thing we really wanted to do was to identify these mysterious muscles. And so to do that, we did whole mount muscle staining at these critical stages from E14.5 when these muscles are first apparent in the hand to E16.5 when they are no longer in the hand. And because all muscles are stained, I've just highlighted for you the relevant ones using the magic of Photoshop. And so you can see at E14.5 are 3 mysterious muscles here. And then by the time we get to E15.5, there's been a dramatic elongation of these muscles down into the arm, coupled with retraction on the distal ends, such that by the time we get to E16.5, these muscles are now completely located within the arm. In contrast, the intrinsic lumbrical muscles, which you see interspersed between these muscles, remain obviously completely intrinsic within the hand. And so this, so if you now look at what is happening to this small tendon fragment, and so this is just a sagittal section taken through the limb. You can see at E14.5, right, we have the muscle in the hand attached to that short tendon fragment, which you can see with the ScxGFP. And then at E15.5, as these muscles are well on their way now out of the hand, crossing the wrist, you can see coupled to that is growth of the superficial flexor tendon, which is still attached to the muscle. And so taken together, this data really suggested that our mystery muscles are in fact the superficial flexor muscles. And now that really opened up kind of a new host of questions because while there are examples of muscle progenitors that kind of move around in the body, so famously the progenitors that migrate from the somites into the limb to generate your limb muscles, or even a few examples of more differentiated single nuclei myoblasts that move from here to there. To our knowledge, there's really no example of multinucleated myofiber movement on the scale. So we did a few things to try to really show that, yes, these muscles are actually moving, which I won't get into. But when we were trying to get this paper in, the reviewers were like, no, this data is not sufficient. We really need you to directly show us that these muscles that end up in the arm were the same ones that originated in the hand, right? And so this was difficult to do because there are ways to genetically label tissues and then trace the fate of those tissues. So for muscle, we could have used a PAX3 Cre or a PAX7 Cre to label muscles, but then all muscles would be labeled. We needed something that would only label the superficial muscles, or what we think were the superficial muscles. And there's no gene that would distinguish really one muscle from another. So after puzzling over this for a little while, we spoke to one of our collaborators at OHSU, John Brigandy. And what they do routinely in their lab is transuterine microinjection. So they can directly inject anything you want really into developing embryos, and then the embryos continue to develop. You can see what happens later. And so we did this experiment where we took a Cre reporter mouse, and for those of you who are not so familiar with the Cre lock system, basically, all cells in this mouse have the potential to express a red fluorescent protein, tomato protein. However, they don't express it because of this transcriptional stop that's inserted right before tomato. In the presence of Cre recombinase, however, it will recognize these LOX-P sites that are flanking the stop and delete it out. And so now the cells that have been exposed to Cre will be able to express tomato, and in our case will be labeled red. So how to introduce Cre only to the muscles in the hand? And so with this, we were able to rely on John to inject for us an adenovirus that encodes Cre recombinase. And so adenovirus infection is non-specific, but we can target the adenovirus just to the hand to label the tissues of the hand. And if later we look and the muscles are indeed labeled, then we would know that they originated from the hand. So, right. So we did this experiment, and this is what we found. And so this is just a bright field image of one of these injected limbs. And this is the same limb, but now looking at the red channel to look at these labeled red cells. And you can see there's, quite a bit of labeling. And in fact, in the arm, it looks like there might be labeling of the superficial flexor muscles. So taking a section through the hand, or the paw, you can see that there's a lot of non-specific labeling of tissues. Not only are the intrinsic muscles labeled, which you can see by myosin staining, but a lot of other tissues, tendons, blood vessels, undifferentiated mesenchyme, the periostic, like lots of labeling of the hand tissues, which you would expect. But now when you get into the arm, even though there are many other muscles, obviously in the arm, many other tissues, now what we only really see is labeling of the superficial flexor muscles along with its associated blood vessel, suggesting now directly that these muscles really originated from the hand at E14.5. And then so with that, we were able to get the paper in, and it was very exciting. Usually, I feel like things don't work that well. So now having identified that, yes, the superficial flexors form and differentiate in the paw and then translocate out of the paw into the arm, the next question was, why is this muscle doing this at all? It's very specific to this muscle. It's unique, and in development, there are actually not so many examples of this kind of correction. So when we do see that, it usually indicates that maybe there's some underlying reason for it and something kind of interesting going on. And then so we start to do some just comparative anatomy, and what we found was that in humans as well as mice, the superficial flexor is only found in the forelimb. In the hind limb, we don't have superficial flexors, but we do have what seem to be anatomically related tendons and muscles called the flexor digitorum brevis. And so the tendons of the brevis are very similar to the tendons of the superficial flexor, but of course you can see here that the muscles of the brevis are completely intrinsic within the foot. So now we were sort of interested in looking at what happens to brevis development in the mouse during these critical stages, and we actually found several similarities with that of the superficial flexor. So this is now showing sections of the brevis. Like the superficial flexor, it also develops late relative to the other tendons in the foot. And then it also undergoes this sort of dramatic elongation of the muscles. However, while the superficial flexor obviously continues to translocate out of the hand into the arm, the brevis elongates but then remains completely localized within the foot. So they seem to be homologous muscles based on their developmental progression. And then so while mice and humans have superficial flexors in the forelimb and a brevis in the hind limb, other mammalian species such as dogs and horses, and indeed from what I could tell, all hoofed mammals, so ungulates, they have superficial flexors in both the fore and the hind limbs. And so, in fact, when I was sort of searching through all the animals I could think of, or the mammals I could think of, in no case could I really find a brevis muscle in the forelimb, except for very, very primitive species such as egg-laying mammals like the platypus. And now if we extend our search wider to look at our ancestral tetrapods ancestors, so amphibians, for example, now instead of seeing a superficial flexor, we actually see brevis muscles in both the fore and the hind limb, and now in no case could I find a superficial flexor. So taking this all together, the reason, or kind of like rationale for explaining this odd progression of superficial development is that formation of the superficial flexor is a modification to the developmental program, where the original program was really formation of a brevis muscle, and then translocation of this muscle out of the limb is a later evolutionary event. And so in some cases, such as in humans or in mice, this translocation only happens to the forelimb. In others, there were two events where it happened in both the fore and the hind limb. And so the developmental process was recapitulating this evolutionary history. And now, we're trying to think, oh, could this be relevant or interesting clinically in any way? And I guess I started looking for just variations and maybe pathologies associated with the superficial flexor. And I was able to find many cases sort of summarized here in this review of ectopic muscles associated with this tendon found in the hand, muscles that extend too far into the wrist, or extra muscles kind of coming off the tendon. So a lot of sort of natural variation in the population, and in some cases this resulted in hand or wrist pain, which is how they were discovered. And then so we also have evidence now to show that, and this is not something we generated, it was referred to us by a collaborator, that this developmental process likely happens in humans as well. And so this is a section taken from a human fetus showing 4 superficial flexor muscles in the hand at this early developmental stage, and they are later replaced by tendons. And so in cases where we do see these like sort of interesting anomalies in the superficial flexor, to me, knowing what we do about the developmental process, it really suggests that these variations represent some failure or partial failure in movement of these muscles out of the hand. While we have many mouse mutants where we see sort of complete failure of the muscles to move, obviously that's not representative of the human population, but we also, in some of our heterozygous mutants, find cases where we have sort of these ectopic muscles associated with the superficial flexor tendons in the hand, indicating that where this kind of variation might result in some kind of hand pain, that there may be a genetic predisposition in some individuals for these syndromes. So I mentioned my postdoc lab was a tendon lab, and you wouldn't really know it by all this time I'm talking about the muscle, but remember that as the muscles are forming and then sort of moving out of the hand, the superficial flexor tendons are also being formed at the same time. And so now I'm going to shift gears a little bit, stop talking so much about muscle, and now focus on the tendon part of this process. Although there are some genetically distinct aspects of superficial tendon formation relative to the other tendons, conceptually the way these tendons form is the same as all of the tendons of the limb. So here is back to our wild-type schematic of an E14.5 limb, where you have the short tendon fragment and the muscles. And then what we find is that formation of the tendon is actually modular, meaning that each piece can be formed independently of the other piece. So, so while eventually these end up being obviously continuous structures coming from muscle to skeletal insertion, in the absence of signals from cartilage or specifically joint formation, we see that the digit parts are missing in the absence of signals from muscle. So in cases where the muscles don't move, for example, or there are no muscles, you see that these proximal segments are missing. And this kind of developmental modularity, where you can have 2 pieces of the same tendon formed independently of another, is true for all of the tendons in the limb. And so in the case of the superficial flexor, that boundary demarcating modularity is at the metacarpal phalangeal joint, but for all of the other tendons, it is actually at the wrist. So now I'm going to really talk generally about the limb tendons, because the stage where that really regulates general limb tendon development is a little bit earlier than superficial flexor, and it's kind of easier to generalize everything. All right, so kind of one of the first pieces of evidence we had that there was this interesting modularity in the formation of limb tendons came from a mouse that we call muscleless. And so this isn't a true muscleless mouse, it's only limb muscleless, but it's a mouse where there is a point mutation in the PAX3 gene such that these muscle progenitors do not migrate into the limb, and you end up with a limb with no muscles. So in the absence of muscle, what you see here is that by E13.5, while this is a wild-type tendon pattern, you can see that when we focus just on the extensor communis tendons, that the hand parts of these tendons are present, but all of the arm parts are missing. And indeed, all of the arm parts are missing for the other tendons as well. What is interesting is that this phenotype doesn't manifest until E13.5. So if you look at one day before at E12.5, you can see that the muscleless mutant is actually very similar to the wild type. So the limb is a little bit smaller, but all of the segments found in the arm are also present, even though muscles are missing. And so this seemed to be a critical stage, the stage between E12.5 and E13.5. And so we're really interested in what was happening during the stage and and where this dependency on muscle arises. So this is going back to kind of this earlier picture I showed here. And so going back to that a little bit, this E12.5 stage, it's a stage where we first see that tendon pattern emerge, but it's also the stage that all of the musculoskeletal tissues actually become integrated. So if you look at what's happening to the skeleton and muscle, for example, at E12.5, and this is a whole map multiphoton image where the skeleton or the cartilage is shown in green and the muscles are in red, you see at this stage the skeleton in the arm is actually extremely short, and there are muscles that span the entire length of the arm. And these muscles aren't just floating around the space, but they're actually connected to short tendon fragments in the wrist. So for the tendons like the commutus that actually extend all the way into the hand, they directly connect to this sort of like organizing center of tendons around the wrist, and this connects to the hand segments. So at E12.5, you have a short arm, muscles that span the length of the arm attached to short tendon fragments. And then between E12.5 and E14.5, what you see is dramatic elongation of the forearm skeleton. And then also in tandem with that, you see similar elongation of these short-range tendon fragments, such that the entire system, the muscles, the tendons, and the skeleton remains fully connected at all times during these stages of development. And so, so now going back to our muscleless mutant, you can see that, all right, so at E13.5, remember you have the phenotype where you don't see the tendons in the arm, and at E12.5 they look normal, suggesting that induction of these short-range tendons at E12.5 is independent of muscle, but that this elongation event really requires an attachment to muscle. And we think this attachment is important because skeletal growth is likely the main driving event here, and sort of the signals to the soft tissues that regulate soft tissue development and growth is a secondary signal imparted by the skeleton. So, so for the rest of this talk, I'm really going to focus on this elongation stage. So what is happening during the second phase of tendon development in the arm, and how cells are being added. So while the matrix obviously becomes a really important component of tendons in later stages, at these very early stages we're talking about, it's really just an addition of new cells. And so cells can be added to the short-range tendon either by proliferation of these original progenitor cells or recruitment of new cells into the tendon to facilitate growth. And so we looked for proliferation during these stages, and using BRDU labeling, we can see, yes, indeed, there's quite a bit of proliferation going on in tendons at these critical stages. And so we are next interested in seeing whether there's also a recruitment of new cells. So there are a couple of different ways you could do this. I'm not going to show everything, so I just want to say that we took a couple of approaches. And so this first approach is traditional lineage tracing, where you label cells at one stage and then see what happens to the cells at a later stage. And so doing this, using a ScxGFP-neg in order to label early tendon progenitors, we were able to have some evidence showing that new cells are indeed being recruited into the tendon to facilitate growth. And this is distinct from other tissues like cartilage, for example, where initially you have an allocation of progenitors and then you have amplification of this initial progenitor pool to form the resulting tissue. For tendons, it seems like there may be a continuous recruitment process where new cells are being added in order to drive growth. And so the second approach is what I will focus on, is again in collaboration with John Burgundy and Lin. So I showed data before where they injected this adenovirus for us in order to be able to label tissues in the hand. And now what we wanted to do is to directly show that cells are indeed being recruited. And so we wanted to introduce a labeled outside population of cells that would not be part of the original short-range tendon to show that they could be incorporated into tendons and that there was therefore recruitment. And so I didn't really get into this before, but the way this works is you stage female, so you know what stage embryos that you're getting. You make a ventral incision in the female to expose the uterus containing all these embryos. And then you can inject, if you can say, into targeted location. In this case, we're injecting labeled cells into an E12.5 limb, so very tiny. And then she puts them all back into the female, and they continue to develop normally for a few more days. So this is a pretty difficult procedure, but we are able to do this several times. And so in order to distinguish the donor cells from the host cells, we genetically labeled them using PRX1-Cre. And so PRX1 is a gene that's expressed by all limb mesenchymal cells. And so if you have an embryo that is positive for this combination, you'll see that their limbs are just bright red. And so we isolate the cells from the limb and then inject them directly into a stage-matched GFP embryo that does not have sort of this reporter in it. And so basically, the donor cells will be red, the host cells will not be red. However, all of the cells will have the ability to express this ScxGFP tendon reporter given the correct cues. And because this is very difficult, not every injected limb will retain these rosa tomato labeled cells. And so to kind of do a first screen, we just inspect by whole mount and only further evaluate the limbs that have a positive signal. And then to define recruitment, we would collect transverse sections all the way from digits to the elbow of these positive limbs, and then scan every section for incorporation of these labeled cells into tendons. And so recruitment was defined as a cell that was in a tendon that was both tomato positive and ScxGFP positive. So using this method, recruitment was not a very frequent event. It was actually fairly rare. However, in the several rounds that we did this, we were able to reproducibly and consistently find labeled cells within tendons. And so this is an example tendon where you can see a couple of cells incorporated into the tendon, and these cells are also positive for ScxGFP, meaning they've taken on the tendon cell fate. Interestingly, proximity does not necessarily induce a tendon cell, so you can see one of these labeled cells sort of directly adjacent to a tendon. However, it's not taken on ScxGFP, and it's therefore defined as not recruited. You can see that generally speaking, there's quite a bit of labeled cells that are around, and again, they're not GFP positive and not tendon. So, so we're able to show now that, okay, so you have this model of understanding how long tendons form in the limb, where there's first induction and then elongation. And then elongation is fueled by both proliferation of cells and also a recruitment of new cells into the system. And so with that framework in mind, we were actually able to make sense of a mouse mutant we had that had kind of eluded us for a few years. And so this is a Skeraxis mutant. So I've been talking about kind of Skeraxis as a tendon reporter for this entire talk. And so it was the first transcription factor that was identified for tendons, and in fact, it is still only one of the very few transcription factors that's been identified, and the only one that is expressed from these early stages onward. And so when this mutant was first generated, we thought that that you would probably get a mouse without tendons, right? So that maybe Skeraxis would be the master regulator of tendon cell fate in the way that maybe SOX9 is for cartilage during these stages. However, you can see here, this is the Skeraxis mutant. Obviously, it's viable. The tendons are affected, but it survives. And importantly, what we found is that the ability for our muscle to attach to a skeleton, so formation of a tendon, was actually not disrupted in these mutants. There was a tendon-specific phenotype, however, and where we saw this is really in formation of the long tendons in the limb and in the tail. Sorry. So formation of short-range tendons was actually fine. It was long tendon development that was really impaired. And so at E12.5, Skeraxis mutants are normal, and the phenotype really first manifests at E13.5. And so in the context of our model now, it really appears that Skeraxis, right, does not regulate formation of short-range tendons. It doesn't impair tendon induction, but what it might actually be required for is tendon elongation, and specifically recruitment, because we see that Skeraxis null tendons do proliferate. And so we now did this experiment where we generated like a chimeric scenario where we deleted Skeraxis only in a subset of tendon cells, and then tried to see what happened. And so this is a wild type limb at a fairly late stage at E18.5. You can see ordinarily it's composed of a mixture of these ScxGFP lineage cells and non-ScxGFP lineage cells, so red and green cells, but all the cells are wild type. And so normally there's sort of this mixed composition, more as you are close to the wrist or the skeletal insertion, and less as you go on, but still a mixture of these cells. And now if you look at our mutant, where now the red cells are not wild type, in fact they're ScxGFP mutant, what you can see is that now as you go from the wrist and down into the tendon mid-substance set, these tendons are now composed exclusively of the wild type cells, so they fail to recruit mutant cells during the process of elongation. This was even more striking when you look at some of the tendons in the tail. So ordinarily, dorsal tail tendons are highly composed of these red wild type cells. However, in our mutant, again, now these tail tendons are completely exclude these red cells, which are now mutant, and in fact, they are almost seem to be surrounded by the red cells, as if the red cells maybe were trying to recruit, but then were not incorporated. And so in certain regions of tendons, very close to the wrist, for example, you can see mixed populations of red cells forming around tendons, but these red cells seem to have really lost a lot of their ScxGFP expression, or perhaps they never had it. And you can see that you see an interesting segregation of wild type and mutant cells, suggesting that ScxGFP may be regulating recruitment by way of some kind of adhesive molecule. And so in the absence of ScxGFP, cells are not able to be recruited because they don't express this molecule. And we're currently screening to see what that molecule might be. So just to summarize sort of our model for understanding tendon development, first, we have induction of a short-range tendon that is directly connected to muscle. And then the second phase of growth is really this elongation of this short tendon fragment that's driven by a combination of proliferation of these original cells and the recruitment of new tendon cells. And so while ScxGFP is not required for induction, we find that it is required for elongation, specifically of recruitment. And so because we found that tendons in the end were composed of cells that were positive for ScxGFP, it suggested that ScxGFP was required in the cells that were being recruited in order to incorporate. So I just kind of briefly went over these two stories from my postdoc, and now, so one of the things about, I guess, like a research career is that you do training as a PhD student, and then you do training as a postdoc, and eventually you're expected to really sort of take what you've learned from there, but distinguish yourself from your previous mentors. And so obviously my postdoc advisor, Ron Schweitzer, is continuing to do work on tendon development. And so where I thought I would take my own research is to study similar questions, but in a different context, and probably in a more clinically relevant context. And so I'm really interested in understanding stem cell biology and differentiation during healing and regeneration in postnatal stages. So tendon biology overall, there's very little that's known, and even though there's been a lot of sort of focus and emphasis on understanding tissue healing, especially in adults, and scar formation, the basic biology of these processes is still kind of largely a black box. And so the questions that I'm interested in focus on sort of molecular regulation of tendon sulfate. In the postnatal stages, this would be sort of activation of stem cells and recruitment of stem cells. The factors that drive tendon differentiation versus fibrotic scar, and I would expect, or I hypothesize that likely it's triggering different cell populations under maybe inflammatory conditions. And then to sort of, and so that's just kind of the general scope of where I'm hoping to take my lab. And then so using kind of the genetic mouse mutants and models that we have to study embryonic tendon development, we can now apply them to look at what happens during healing and regeneration. So there have been stem cells are thought to be in tendons, however, they have not been well defined. And so Mary and Young's group was the first to isolate such cells from tendons. They were able to show that they are multipotent and self-renew, so they had characteristics of stem cells in vitro, but there's really no clear sense of where they might be in the tendon or near the tendon or sort of distinguishing markers for these cells. And so they suggested using kind of an indirect approach that these cells may actually be intrinsic within the tendon. Others have shown, however, that progenitor-like cells may reside outside of the tendon proper, so residing in the basement membrane epitendon layer that surrounds tendons. And these cells can be, it seems like they could be activated by exercise and migrate into the tendon, or triggered by injury. And so Nat Dement, when he was a graduate student with David Butler, showed that in an adult patellar tendon injury model, that these cells, these external paratendon cells, are activated and they kind of form a band-aid over the defect. And these are in adults. So so that's kind of my first step, kind of, was to try to identify more definitively the cells that are actually participating in the healing process. And then so I am using a couple of different inducible Cre lines. So I've described the Cre lock system, but what we can also do is have inducible Cre lines where the Cre recombinase is not active in the cell until you introduce tamoxifen. And so by doing that, you can now control temporal labeling or deletion of cells. You can target specific cell populations, and so the ones I'm interested in are putative stem cells and differentiated tenocytes. And then look at gene function by way of conditional mutants. So you can now, depending on when you introduce tamoxifen into the animals, you can now really target early events, later events of before or after injury. So this is just kind of a very early piece of preliminary data, and I was focusing on neonatal healing in very young animals. So this is a mouse pup that's only 5 days old. So we create the injury and then just look to see what happens 3 days later. And so this is a simple Achilles transection model, which, from talking to surgeons, is not maybe necessarily the most clinically relevant model, but for kind of identifying stem cell activities in a low turnover tissue like tendon, it was a very easy and accessible tendon and model for us to adopt. So this is just a transection of the Achilles with no repair. And then obviously what happens is you have retraction of the 2 tendon stubs. And at day 3, after the injury, you can see that there's still a very significant gap. And now if you look 14 days after the initial injury, the same surgery, you can see that the original tendon stubs are still very bright green, but now you have formation of what looks to be a new tendon bridge that's formed connecting the tendon stubs. And this is what I'm calling a neo-tendon. And this tissue that's being formed here connecting the tendon is ScxGFP positive. So now, and try to trace maybe a stem cell source. So SOX9 is a marker that's strongly associated with cartilage and skeletal development during embryogenesis, but postnatally, it's also a marker of stem cells. And so stem cells in the intestine, for example, liver, pancreas, hair follicles, many of these cells actually express SOX9. And so I kind of wanted to see whether, I knew there was some labeling of SOX9 cells in tendons that we can get postnatally, and so I wanted to see if maybe this would represent a stem cell population. So this is a very busy slide. I just kind of like copied it over from somewhere else, but basically if you introduce tamoxifen to label SOX9 expressing cells at P2 and P3, create the injury at P5, you can see what happens to these initially labeled cells later on. So even if the cells no longer express SOX9, if new cells turn on SOX9, those cells will not be labeled. You're only labeling cells that express SOX9 at this stage. So in the normal situation, there's sort of sporadically labeling of SOX9 positive cells and tendons. In the injury, in the injured limb, however, you see a strong activation of these cells in the original tendon stubs and also in the forming neo-tendon, suggesting that when triggered by injury, these cells that are usually sporadic may amplify and become this population that then gives rise to the neo-tissue. And so I'm trying to study this further, look at different stages, and also to test whether they may be an intrinsic cell population by using an inducible ScxGFP. So this is just some very early data that I thought would be fun to share with you. And so and I'm not going to go to all of this, but basically we are also on the side, also developing an in vitro system to study tendon differentiation, and this is really useful mostly to look at gene regulation in a more controlled environment, and then also to screen for new inducers of tenogenesis. Because right now, really, there are only a very small handful of molecules that have been shown to induce tendon differentiation. What I am interested in doing is to screen maybe siRNA libraries or small molecule libraries to find new factors, and then to maybe open up new signaling pathways that are important for tendon differentiation. And so we're working on miniaturizing this system with a collaborator. And then moving forward, again, not getting into all of this, these are sort of the main projects in the lab that we're really interested in going forward. So some of the neonatal healing compared to adult healing, I showed a little data for that. And then also focusing on the non-canonical TGF-beta signaling pathways compared to SMAD-mediated pathways to try to differentiate the tendon differentiation activities of TGF-beta signaling versus scar formation. And then lastly, a project that we will develop in time is to understand how mechanical regulation might influence tendon differentiation during healing. And some of the inspiration from that comes from some bone studies. Okay, so with that, I think I'm going to end. I'd like to acknowledge my postdoctoral lab and advisor, Ron Schweitzer. He's fantastic and amazingly supportive. Some of you will know him. But I also really wanted to acknowledge kind of my new family here at Mount Sinai. So obviously too many people to name, and I think that's James. I don't even know. That's definitely Nellie and Evan. But everyone's been so fantastic in helping Kristen and I get settled and feel at home here at Sinai, and so incredibly helpful. And I'm excited to meet some of you as well. So thank you.